easyGDB also provides a tool to visualize gene expression data. We can enable this tool very easily in the configuration file, and to use it we just need to provide a list of genes and the dataset we want to query. Datasets are just text files that we can place in the expression data folder, and their format is just a tab-delimited file that we can easily obtain from the R analysis just by normalizing the replicates. The output is very interactive. The results are organized in different ranges of the expression scale that we can filter. Additionally, we can also change the color of the results, and we can download this output. Another graph allows us to explore the values of the replicates of each one of the genes. And a table that can be downloaded in many different formats shows us the average expression values.